that's a very innovative study that we launched and started to do in patients with myelofibrosis, currently as an add-on approach to stable dose of Ruxolitinib. There were some first data that came from using regulatory T cells in patients with inherited bone marrow failure syndromes, which also included patients with myelofibrosis. And then we were kind of encouraged by seeing responses in the cytopenias. So it was taken into the phase 1b study of exploring specifically regulated or engineered Tregs that have CXCR4 enrichment, so they could home to the bone marrow. And then the 6 infusions added into patients that have Ruxolitinib for myelofibrosis every month, 100 million cells in every single bag. And then there was a phase 1 study, so we were looking for safety. It's mostly to tackle the inflammation and to see whether we can somehow challenge the microenvironment. Regulatory T cells are the gatekeepers. They pretty much are playing with the bad players and the good players and somehow regulate the disbalanced immune system in patients with various diseases. For example, use an anti-immunity diseases, neuroinflammation. So that's MPN, because particularly myelofibrosis, but also other MPNs are kind of a hallmark of inflammation. That's why we thought that the Tregs, they are being brought to these patients from either healthy individuals or cord blood, as this study has, would help to regulate and navigate the dysfunctional immune system and maybe enhancing the efficacy of the current therapies, or even help us with the progression. So there are data from previous studies showing synergism with Ruxolitinib that could also be employed in preventing infections or adverse outcomes that could come with the therapy, specifically more immune dysregulation, such as impact on NK cells, Tregs, or even innate immunity. So that's why the rationale for using the regulatory adaptive T cell therapy came from.